protein purification. So I'll be talking about the idea, Chris will talk about the plasmids that we designed. Jeff and Doug will talk about what we actually built and the data that we got from it, and Joseph will be talking about what we still have left to do. So what's wrong with traditional protein purification? Why is it so bad? Well, Jeff here does a lot of protein purification in his lab, and he knows that it really sucks. It's a really long, boring process. First, Jeff clones his protein, or a favorite protein, into an expression vector, grows up his cells to produce the protein, then lyses, pellets, and filters the cells to separate the soluble and insoluble protein from the cell fragments. Then he loads the protein onto a column to bind only the protein that he wants, his protein of interest. And then finally elutes to give his purified protein. Well, this takes Jeff about 3.5 hours, and he's pretty good at the process. 3.5 hours of bench time, and he's pretty good at the process. So we were thinking, how can we make this better? Well, how about we use the cell as some of these steps? So, for all these steps, the whole point of these steps is to remove the protein that we've just expressed and move it into the media. So, that sounds a lot like a secretion system. So let's just use a secretion system here. And then these steps, all we're doing is separating our protein from all the other protein that we've released from the cell. So why not use the cell itself as a column? So what would this look like? Well, again, you put the protein of interest into an expression vector, now the target vector, which will express your protein now as a fusion protein with 2 tags on it. The first tag, a secretion tag, interacts with a secretion system that we put in the cell to secrete the protein outside into the media. The second tag, a display tag, will bind the cell to a surface display protein that we're also displaying on the surface of the cell. Finally, we can release the protein, similar to the way that you'd release protein off the column, by substituting in, by eluting with a small molecule that will displace the protein.
It's the secretion plasmid. We got this type 1 secretion system from Orwinia chrysanthemi. It consists of 3 parts, P, R, T, D, E, and F. We maintain the native ribosome binding sites to keep the levels of each part relative, and we put it in a low copy number plasmid to not give too much dosage to the cell. So the way this works is PRT, D, E, and F work together to form a core that goes all the way from the cytosol to the extracellular fluid, which is one of the advantages of this secretion system. The other reason we chose the secretion system is because it's been shown to secrete lots of proteins of different sizes, and because it's selected for proteins that have PRTB on them. Finally, the last part of our system is the display plasmid. This was a part that was already in the registry, submitted by Harvard in 2006. And they had 4 different kind of flavors on the same idea. So what they did is they had a fusion protein that had LPP, which is a signal that sends the fusion protein to the periplasm, AMPE, which then anchors